Welcome guys, in today's video we'll be learning about cellular respiration. What is it? It is the process by which organisms use oxygen to break down food molecules to get chemical energy for cell functions. So if you remember in the previous topic we talked about photosynthesis, and in photosynthesis we took simple molecules and made it up into complex molecules. And the complex molecule we made in photosynthesis, well not us particularly but the plants they made was glucose, which was food. So in respiration, we take that food and break it down into simple molecules where we can then use the energy that we got from the simple molecules for the cells to do their functions and whatnot. There are two types of cellular respiration. There are aerobic respiration and there is anaerobic respiration. Aerobic means oxygen, so aerobic respiration is in the presence of oxygen, and anaerobic, and meaning no or not, and aerobic meaning oxygen, no oxygen, is in the absence of oxygen. Cellular respiration takes place in the mitochondrion. Remember, one mitochondrion, many mitochondria. It is a site for cellular respiration. The mitochondrion is an organelle found in the cytoplasm of a cell. Here we have a label diagram of the mitochondrion. We have small green dots are ATP synthase, red dots are granules, the blue small dots are porins. You don't need to know the first three. Then we have the intermembrane space. You have your intermembrane, which is basically your crystal, your membrane folds. And you have your outer membrane. And then you have your ribosomes, and your mitochondrion, DNA, and you have the matrix. These are the labels you need to know. Remember, I said in the previous topic that we have a fluid-like substance in the cell called the cytoplasm, and in the chloroplast it is called the stroma. Now in the mitochondrion, you have the matrix. That's the fluid-like substance found in the mitochondrion. Cellular respiration can be written in a chemical formula. Our reactants or requirements are C6H12O6 and 6 oxygens. The C6H12O6, if you could remember, that was the glucose which we made in photosynthesis. So those glucose are broken down, and when we break it down with the help of oxygen, we get carbon dioxide, 6 carbon dioxides, 6 water molecules, and ATP. ATP is the energy that we require for the cells. So we have the requirements and the reactants, and all that we get our products. Here we have a table showing cellular respiration versus photosynthesis. The location for photosynthesis is chloroplast, and the location for cellular respiration is mitochondria. The type of reaction for photosynthesis is an anabolic, where simple molecules are made up into complex molecules, and in cellular respiration we have a catabolic, where the complex molecules or substance is broken down into simple substances. The reactions for photosynthesis are carbon dioxide, H2O, water, radiant energy, and chlorophyll. So these are requirements basically. And in cellular respiration we have C6H12O6, which is glucose, and oxygen. The hydrogen carrier for photosynthesis, remember, was NADP. NADP stood for nicotinamide adenine dinucleotide phosphate. This is which the hydrogen areas, the hydrogen energy-rich hydrogen atoms, which we got from the splitting of the water molecules, they were carried by these enzymes. They form the coenzyme. And cellular respiration we have NAD, which is just nicotinamide adenine dinucleotide. Photosynthesis it occurs during the day and not at night, because remember we require radiant energy to undergo this process. Cellular respiration occurs day and night, so it occurs throughout the day basically. It doesn't stop. So how does cellular respiration work? Cellular respiration can be divided into three main groups: glycolysis, the Krebs cycle, and oxidative phosphorylation. Now remember we mentioned there's two types of cellular respiration. There's aerobic and anaerobic. One occurs in the presence of oxygen, the other one occurs in the absence of oxygen. Now glycolysis is an anaerobic respiration. It doesn't require oxygen to undergo its process. But the Krebs cycle and oxidative phosphorylation, they do require. So glycolysis is basically the first step to anaerobic respiration. And I'll show you why. Here's the basics of the bonds between elements and what happens between them and stuff. So we have a bond between carbon and hydrogen. Remember, we want to get energy, alright? We need energy for our cell functions. Hydrogen is high in energy. Hydrogen is basically an electron that has lots of energy. So, how do we get it? Dehydrogenase. It is an enzyme, right, that catalyzes the removal of hydrogen. So what it does is it speeds up this reaction between the hydrogen and the carbon to that when it separates, when it breaks the bond, and then when the hydrogen is broken off from the carbon, it now binds to the hydrogen carrier called NAD, nicotinamide adenine dinucleotide. And then here we have two carbons bonded to each other. The enzyme that catalyzes the removal of carbon is decarboxylase. So decarboxylase, the enzyme, catalyzes the removal of this. It speeds up the reaction of removing the carbon bonded to another element or another carbon. And here we have ADP, adenosine diphosphate, so it's adenosine bonded to two phosphates, bonded to another phosphate, which makes it ATP. So this whole thing is ATP, adenosine plus the phosphate. Now there is a bond between the phosphorus, and in that bond there's energy. The energy is stored in this bond. So remember, if you create a bond between the two, energy is stored. But when you break the bond, energy is released, because remember there was energy in it. Okay. The first step of respiration is glycolysis, which is an anaerobic respiration, type of respiration. So remember, what was our reactant? Our reactant was glucose. We are breaking down the glucose to get the energy found in it, so that the cell can use it for its other functions. So to break it down, we require activation energy. So the activation energy is the ATP molecules found in the cells. So here comes along two ATPs. Now remember the bond between the phosphates, there's energy stored in that bond. The bond now breaks, right, and energy is released. That energy that was released, it is used to bond two phosphates to the glucose. So remember, there's two ATP. The bond breaks. We're left with two ADP and a P that's just separate. There was energy that was released from the removal of that phosphate, the breaking of the bond. That energy is needed. Remember, that activation energy was needed to take the two phosphates that were separated from the two ADP and bond it to glucose. And then it resulted in glucose 1,6 diphosphate. So we have glucose, and phosphate is bonded to the first carbon and the second carbon. So we have 6 carbons. We have 1, 2, 3, 4, 5, 6. There's a phosphate attached to the first and a phosphate attached to the second. The last one, the 6th one. And it's called diphosphates because there's 2 phosphates. This now creates the glucose 1,6 diphosphate is now unstable. And when it's unstable, it splits in half into 2 identical parts. Then we left the C3 compound. So it's going to be 3 carbons on this side and 3 carbons on that side. And remember when we said if you have a C3 compound, right? 2 C3 compounds, and they have energy in it, and there's a phosphate on it. That PJL, or a C3 compound, is known as PJL, which is a phosphoglycerol acid. No, phosphoglyceroldehyde. Phosphoglyceroldehyde. And now we have two C3 compounds. Now remember the enzyme dehydrogenase. It catalyzes, meaning and it accelerates the removal of a hydrogen. So it removes the hydrogen from the C3 compound. And remember, whatever we do on this side, we're doing on the same on that side. So whatever we do to the C3 compound, it happens to that C3 compound. But I'm going to explain from this side. So we remove now the hydrogen. We remove two, because here you can see there's two. So here comes the enzyme NAD, comes along into the function or this process, and it picks up the hydrogen atoms that were removed. Remember, these are energy rich hydrogen atoms. We're gonna use this energy rich hydrogen atoms later on, and that's gonna be using oxidative phosphorylation. So now the coenzyme NADH2 goes to the oxidative phosphorylation stage. Okay, so we remove two hydrogens. Now remember, when you remove two hydrogens, right, you break the bond. In that bond there's energy. So when you break a bond, energy is released. That energy that was released is now used to bond two phosphates to 2 ADP, and it resulted in 2 ATP. Remember, we wanted energy in the form of ATP. If you remember your chemical equation, one of the products was ATP. We wanted that energy. So here we have some of that energy. So now we have created 4 ATP in total, because here's 2 here and here's 2 here. And we used 2 in the beginning. So we have a net total of 2 ATP. Remember, your net total is your the amount that you produce minus the amount that was used. So produced minus used, and we get a net total of 2 ATP. Okay, so we remove 2 hydrogens, right? And now we're left with PGA. PGA is just your C3 compound with a phosphate to it. Now that PGA, the phosphate is now removed from the PGA, right? And no energy or little energy is required to remove it. And now we have a pyruvate. So in total we have 2 pyruvates. And pyruvate is CH3COCOOH. I don't know why they wrote it that way. I don't know why they couldn't just group it. Maybe that's just how it's bonded like how it's bonded. So they wrote it the way it was bonded. I'm not sure, but yeah, I think so. I think it's like that. Yeah, it is like that. It is like that. It makes sense. So in total we removed 4 hydrogens. We produced 4 ATPs, and we had a net total of 2 ATPs. A phosphate was removed. That phosphate is probably going to be used in another function somewhere else in the cell. And remember the 2 ADP over here, they're also going to be reused again. So we left with 2 pyruvate. Now let's see if the math works. In the beginning we had how much did we have? We had 6 carbons, we had 12 hydrogens, and we had 6 oxygens. Now after we remove those, let's see if it adds up here in the 2 pyruvates. So we remove a total of 4 hydrogens. How much hydrogens? If we remove 4 hydrogens from 12, we should have 8 in total. So here we have 3 over here and 1 over here. That's 4 on both sides. Then we have 8 in total left. So it adds up. We removed no carbons, so we should have 6 in total. So here we have 1, 2, 3 here, and 1, 2, 3 here. So we have 6 in total. That's correct. And oxygens, we had 6 oxygens. Here we have 3 over here, and we have 3 over there. We have 6 in total. So the math does add up. So now we left with 2 pyruvates, and this is going to be adjusted before we take it into the next stage, which is the Krebs cycle. So we have a pyruvate, right? But if we do the one between the other, this is just showing one pyruvate. Yeah, it shows the chemical bondage between the carbons and the hydrogens and the oxygen. So this is before it goes into the Krebs cycle, which is in fact where it takes place in the cristae, the membrane phones of the mitochondria. So we have the enzyme called dehydrogenase, and it catalyzes the removal of hydrogen. Remember, hydrogen is energy rich, and we're going to use that in oxidative phosphorylation to get more energy. So here comes NAD, the hydrogen carrier, and it picks up the hydrogen that was removed, and that hydrogen is taken to the oxidative phosphorylation stage. So this coenzyme is going to there. Now, when you remove hydrogen, energy is released. That energy that was released is used to bond a ADP to a P, resulting in a ATP. And now we have another enzyme called decarboxylase. Decarboxylase catalyzes the removal of oxygen, right? So you see over here this this carbon over here, bond now breaks. Okay, this bond here, it takes these two oxygens and it's gonna be liberated. It's gonna be freed out of the cell basically through gaseous exchange, diffuses into the blood, and then we breathe it out, and so on and so on. So this is where the oxygen is required. Remember, we have two types of cellular respiration. We have aerobic and anaerobic. This is the aerobic part. We require oxygen because oxygen is a carrier for the carbon. So yeah, the carbon is now liberated, and we are left with acetal. So acetal is CH3 with one carbon and one oxygen. So in total we have two carbons. So you see the bonds here between the carbons and the oxygen. So now we adjusted the acetal. Now this acetal can finally go into the Krebs cycle. Remember, we still haven't started with the Krebs cycle. This is like an intermediate phase. So this is after glycolysis but before Krebs cycle. So it's an in-between phase. So we have an acetal, and an enzyme comes called coenzyme A, and the acetal attaches itself to the coenzyme, and the coenzyme transports it to the Krebs cycle, which is in the cristae of the mitochondria. So this is a C2 compound. There's two carbons in this compound. Remember, like in photosynthesis, we mainly focus on the carbon, even though there is other elements in here. There is hydrogen, there is oxygen, but we don't focus as that much on it. So we're now in the cristae. The coenzyme drops off the acetal, and it goes back to where it started to undergo another carrying process basically. The C2 compound combines with the 4-carbon acid, the 4C acid, to create a 6C acid. So here was 4 carbons and here was 2 added together, and we had 6 carbons here. So either it's acetic acid or a 6-cic acid or a C6 compound, whatever, it's the same thing. And now we have dehydrogenase. Okay, it doesn't show here, but dehydrogenase acts on it, right? And it catalyzes the removal. Remember, there is hydrogen in here, and it catalyzes the removal of hydrogen from the C6 compound. And that hydrogen is taken to the next stage, which is oxidative phosphorylation. Because remember, there's energy in that hydrogen. When you remove hydrogen, energy is released between the bond, and that energy is used to bind a phosphorus to ADP, and you result in ATP. Here is more energy now, so this ATP can be used in the body again for whatever. So she will create more and more energy, but it's not enough. We can still create even more. Now we have the enzyme called decarboxylase. Decarboxylase catalyzes the removal of carbon, right? Carbon from a compound, and that carbon is carried by oxygen, either the oxygen in the actual compound or the oxygen that is just when you breathe. Remember, when you breathe in, you needed oxygen. Same in the requirements for cellular respiration, we need oxygen. There is that oxygen in the cell. So the oxygen in the cell carries the carbon out and is liberated. We breathe it out, and we're left with a 5-carbon acid, a C5 compound. Then this first step over here happens again, or the second step, and hydrogen is removed from the C5 compound, taken to the next stage, oxidative phosphorylation. Energy is released. That energy release is used to bond a phosphate to ADP. We have ATP. ATP can be used by the cells for other bodily functions. We have decarboxylase. Decarboxylase catalyzes the removal of carbon. Carbon is liberated by the help of O2, and it is liberated as carbon dioxide. That's why we breathe out carbon dioxide, and we remove another carbon, so we left with a 4C compound, or 4 carbon acid. And the cycle happens again, because remember there was two pyruvates, so this can happen once again, and then we finish with that pyruvate, and it will continuously happen because there's other pyruvates that's going to come from other glucose, and so on. So that is for Krebs cycle. So Krebs cycle is basically the removal of carbon dioxide and hydrogen, and we formed ATP in here. Next cycle is oxidative phosphorylation. So remember, in glycolysis we removed hydrogen, and I said it's going to be taken to the oxidative phosphorylation. And in the Krebs cycle I also said we removed hydrogen. This is also going to be taken to the oxidative phosphorylation stage. So now we have NADH. This is a coenzyme. This coenzyme, right, comes into the oxidative phosphorylation stage, which is in the matrix of the mitochondrion, and the hydrogen carriers here, they drop off the hydrogen atoms. And remember, these hydrogen atoms are rich in energy. They have lots of energy. So here the NAD drops it off. It is in high energy, and it gets accepted by a hydrogen acceptor. Okay. When it's accepted by a hydrogen acceptor, right, it's going to be released again it's going to be separated. And when you separate it, energy is released. That energy that is released, it is used to bind a phosphorus to ADP to create ATP. Okay. And when it was released, it goes to another acceptor, another hydrogen acceptor carrier, right? And it is again released. When it's released, energy is also released. That energy is released is again used to create more ATP. It goes to another hydrogen acceptor. Once again, when it's released from that hydrogen acceptor, energy is released to create even more ATP. Okay. Now, as we go down the scale, right, the level in energy decreases. So in the beginning, that hydrogen had lots of energy. Then it releases some of its energy, created ATP. It still had energy. It released again energy when it was accepted to another hydrogen acceptor. It released that energy, created more ATP. Now it's even lower in energy, but it still has enough to create even more ATP. And then the hydrogen is low in energy, right? But it still has a little bit enough, right? And so, did I mention that to create a third one? I can't remember, but yeah, I got lost now. We created the ATP, and now it's low in energy, right? It is very low in energy. I can't create even more ATP. It is now released from the hydrogen acceptor, and the final hydrogen acceptor is oxygen. So it is carried out the body as water. It is liberated as water in the urine, I think. Yeah, urine. So remember, it's low in energy. It can't create even more ATP. The final acceptor of hydrogen is oxygen, and it is liberated as water. So that was the final stage of cellular respiration, and we created in total about 38, more or less 38. The textbook gives you 38, but the actual answer is between 30 and 40, I think. 30 and 38. No, yeah, 30 and 38, I think. So there's no definite answer, but just say the total amount of ATP created was 38. That was anaerobic respiration. No, that was aerobic respiration. Now we have anaerobic respiration. Anaerobic respiration, it is called fermentation, and it takes place in the absence of oxygen, so it does not require oxygen. What do we remember? What stage of glycolysis? Oh no, what stage of cellular respiration requires no oxygen? That was glycolysis. So glucose is broken down into pyruvic acid as in glycolysis. So glycolysis happens, right? But as oxygen is absent, so there's no oxygen, so no Krebs cycle or oxidative phosphorylation can occur. So anaerobic respiration, only glycolysis occurs. So here you see this diagram. Anaerobic respiration in plants and animals. In plants it's called alcoholic fermentation, but in animals it's called lactic fermentation. So we have the difference between plant fermentation and animal fermentation or anaerobic respiration. So anaerobic respiration in plants or alcoholic fermentation. In plants and yeast, the pyruvic acid and two hydrogen atoms released by glycolysis are instead converted to ethanol or ethyl alcohol and carbon dioxide. So remember in glycolysis, we created pyruvic acid, okay, two pyruvic acids, and we removed hydrogen. That hydrogen and pyruvic acid is used to convert, or not used, it is converted to ethanol and carbon dioxide. Just as in glycolysis, the reactions yield very little energy for ATP formation, but nevertheless, they enable you to live without oxygen. So remember glycolysis, where the net total of ATP was 2, because we used 2 and we produced 4. So the net total was 2. So in anaerobic respiration, we don't produce a lot of energy, but there still is energy being produced. Now in anaerobic respiration in animals, cells such as muscle fibers, cellular respiration can only take place in the absence of oxygen, remember? And lactic acid fermentation then occurs. So remember, in plants we have alcoholic fermentation, in animals we have lactic fermentation. So glycolysis proceeds normally, but the pyruvic acid combines with the hydrogen atoms released by glycolysis to form lactic acid. So the pyruvic acid plus the hydrogen which we extracted or removed combines together to form lactic acid. Then the muscle fibers receive, when the muscle fibers receive sufficient oxygen again, the lactic acid is changed back into pyruvic acid and can then enter the Krebs cycle. So for us to turn the lactic acid back into pyruvic acid, we need oxygen. So once the body receives enough oxygen, or any oxygen at all, it will convert that lactic acid back into pyruvic acid, and it can undergo the Krebs cycle and oxidative phosphorylation. So that's why when you do an exercise and that stuff, and you're not getting in, and if you're not getting enough oxygen, your body will start forming lactic acid, and lactic acid doesn't allow your muscle cells fibers to form as how they normally should, and they can't stretch and contract as how they could. That's why sometimes if you're working out and your muscles start feeling tight in it means there's a buildup of lactic acid and you need more oxygen. That's why people say you need to breathe during your exercises so that you can have a less buildup of lactic acid. So here's a diagram showing anaerobic respiration in plants and animals. So we have glucose that undergoes glycolysis. We have a total of 2 ATP produced, and that is a little energy. Then we left with pyruvic acid. That pyruvic acid together with hydrogen is converted into ethanol and carbon dioxide in the plants. but in animals they combine together to form lactic acid. And that is cellular respiration. Thank you very much.